Ladies and gentlemen, good afternoon. Today, I'd like to explain you about the sex determination using DNA. But I know many of you here are not so familiar, basically archaeologists, so are not generally unfamiliar with DNA analysis. So please forgive me. It's very, very technical paper, and it has published online January 4th in Prof.Noboru Adachi. If you want to know the detail of my presentation, please visit the site and see the paper. Anyways, sex determination is very, very important both in the forensic field and as well as in archaeology or anthropology. And merit of DNA analysis is that we enable to determine sex even from very, very degraded, fragmented bones or something. So I think the problems of the conventional sexing method is basically two. No, I'm sorry. One is the length of PCR products. This means we generally use a technique called polymerase chain reaction to amplify straight amount of DNA to be the level that we can analyze DNA. This is PCR. The length of PCR products are generally long, and the target region is the second problem. In commercial STL kits that are widely used for modern forensic DNA testing is set about 70 to 110 base pairs. This is the length of PCR products. Generally, in very, very degraded materials, the shorter the amplicon length, the PCR products, the better we get the more reliable results in degraded samples. And the next problem is the target region. Generally, in commercial kits, intron, this means a non-coding region of DNA. Intron region of amelogenin. Amelogenin is related to the human enamel, teeth enamel. Intron region of amelogenin gene is used. But some males, especially in Indian or Nepalese or something, some males congenitally lack this region. So this leads to misclassification of sex. So let me explain our new method of sexing and its features. First of all, we targeted exon region. Exon 2 is analyzed for stability because this region, this coding region, is highly conservative in any races in human. So exon region is targeted. This is the female sequence of exon 2, and this is a male sequence of exon 2. The different bases are shown in yellow letters. This is the difference between male and female. So if we can detect these mutations or differences, we can tell which sex the sample belongs to. And we targeted this base because cytosine and guanine the difference, its construction of base is totally different. This can be assumed as transversion, similar to transversion mutation. Transversion site is chosen to avoid misclassification of nucleotide caused by deamination that is frequently observed in ancient materials. And the next feature is the technique that can identify the base changes. We developed the technique called amplified product length polymorphisms. Very, very technical, I'm sorry. But we design the sequence-specific primers that is specific to female sequence and as well as the male sequence, using the complementary chain sequence. And we designed common forward primer to amplify the region that is between the common forward primer and a sex-specific reverse primer. Then we get the amplified product shown in yellow letters. The length of each product as it stands, both are 56 base pairs. That is very short, but there is no difference. To determine, to identify the sex, if we electrophorese these amplicons, the length is the same, so we cannot identify which chain it is or something. So, to make the difference between two amplicons, we add inosine, 4 bases of inosine to the Amelogenin Y male-specific primers. Then the amplified products for male is now 60 base pairs. So we can detect the SNPs as the difference of amplicon lengths by electrophoresing these amplicons. If you want to know the detail of the merit of inosine addition to the 5' end of each primer, please refer to the paper also published in PLOS One 2 years ago. Then the last feature of our method is the bidirectional analysis of SNPs. Using what you can see here is the sense chain sequence of male and female exon 2 of amelogenin gene. But actually, there are two chains because DNA is, as widely known, DNA is double-strand structure. So there is another chain for another complementary chain for both sequences. Now we use the another chain for designing another set. Again, it's the same amelogenin X-specific and Y-specific primer, and you're adding inosine to the amelogenin Y-specific primer, and then design common, now not forward, common reverse primer to amplify their sequences. Now, here. And moreover, to confirm the male sexuality, we designed we use sex-determining region Y sequence to confirm the male sexuality, along with the amelogenin gene. And the PCR condition, very, very technical, I'm sorry. PCR condition is shown here. The feature of this protocol is annealing extension time of 5 cycles is extended to 5 minutes. It's extraordinarily long to maximize the chance of annealing of the primers to the trace amount of template DNA. And electrophoretic condition is like this, very, very technical. I'm sorry. And this is the sequence of the primers and the amplicons or something like that. Then we move to the testing, the performance of the present method. Now I'd like to show you the detection limit of each set, the primer sets using sense chain, and the primer sets using anti-sense chain. Both are, in both sets, the detection limits is like this. This is 10 picogram, and this is 20 picogram. Both are the same. It's quite very, very sensitive. Much more sensitive than commercial STL kits. And let me show you the robustness test by using 1 nanogram of UV irradiated DNA. UV can damage DNA cut into very, very fragment DNA. So the sense primer sets stands up to 5 joule irradiation of DNA, and in male 1 joule. And the next antisense primers both are stood up to 1 joule irradiation. But when you compare this result to the commercial DNA testing kits, this is very widely used PowerPlex ESX17 fast kit. It can stand up only up to 0.5 joule, and as for female, and 0.2 joule for male. And this is the latest version of PowerPlex kit named PowerPlex Fusion. This kit is better than PowerPlex ESX, but it can stand up only 1 joule for female and 0.5 joule for male. And the result is, the summary of the result is shown here. So as you can see, our method is proved to be much more robust than commercially available STL kits when UV irradiated DNA is used as template. And finally, I'd like to show you the results by using archaeological actual specimen. 14 skeletons excavated from the Funadomari Jomon site. This island is located in the northernmost part of Japan, where Hokkaido University and other joint university teams are now ongoing excavation every year. 14 skeletons excavated from the Funadomari Jomon site, Raven Island, Hokkaido, Japan. It is about 3,500 years BP. These skeletons whose sexuality were securely determined by morphological analysis were chosen. So the results is shown here. By using sense primer sets, we can identify there 9 out of 14 samples are correctly sexed. By using antisense primers, the number is the same. 9 out of 14 samples were correctly sexed. But the contents of the samples are different. As a whole, we can identify 11 out of 14 samples. But by using commercially available STL kits, only samples 7 and 9 could be correctly analyzed by PowerPlex ESX17 fast, and the samples 7, 9, 12 could be analyzed by PowerPlex Fusion. This is the latest version, very, very widely used in forensic field. So now I'd like to wrap up my presentation. New multiplex APLP system for the sex determination of highly degraded samples was developed. This system was proved to be much more robust and sensitive than commercially available STL kits. This system is considered to be suitable for the sex test of ancient human skeletons. That's all. Thank you for your attention.